faster than the 1,000 Daltons. So in fact, the PCR product should be about 800, somewhere around 800 Daltons. So it should run a little faster than this one kilodalton marker. So this is your PCR product that you want to cut out. What are these? PCR primers. You don't want those. So we're going to cut this band out into a pre-weighed tube, and then we are going to pre-weigh the tube, and then you're going to put it in there, you're going to weigh it again so you know the weight of your gel slice. Right, and so then you're going to add 3 volumes of QG buffer. What does that mean? Well, let's say your gel slice weighs 300 milligrams. Okay, so if it weighs 300 milligrams, then we will add 300 microliters of QG buffer, right? So, but you add 3 times that. So you're going to add 3 times 300 or 900. Okay, so for a 300 milligram gel slice, you're going to add 900 microliters of buffer. Okay? Alright, so you're going to do that. Then you're going to you're going to cook that at 50 degrees for 10 minutes. I've got a little hot plate that always stays on at 50 degrees. And then to that, you're going to, once it's dissolved, it should be yellow. Usually it's yellow, so it shouldn't be pop. But to that, you're going to add a total volume of isopropanol. So let's go through that calculation. You had a 300 milligram slice, and you added 900, basically you added 900 milligrams of water, all right? So that means you had a total of 1.2 milliliters that you already have in that tube. 1.2 milliliters. So we're going to add another 1.2 milliliters of isopropanol. Okay? So you're going to add that in there, you're going to stir it up with the vortexer, and then you're going to add it 0.75 mils at a time, because the little column only holds 0.75 mils. Take one of those little purple columns out of the QIAGEN box, and then we're going to add 0.75 milliliters of your yellow isopropanol mixture to that. Put it in the little collection tube, spin it down, be sure to put a balanced piece. Use a clean isopropanol tube that has, you know, like 1 milliliter of water in it. That's good enough as a piece to balance the QIAGEN tube. Okay? You don't need to use a second tube. Those things cost a lot of money. So don't use 2. Just use one QIAGEN thing. Okay. So then you're going to have to spin it down, the flow-through liquid, throw that away, put it back in with the purple column, and then add another 0.75 milliliters. And keep doing that. Spin it down, throw away the liquid, add more. Spin it down, throw away the liquid, add more, until all of your stuff in that yellow liquid with the isopropanol in it is gone. Okay, it has to all be, and what are we doing there? We're adsorbing our DNA onto that little QIAGEN adsorption column. Okay, so discard that flow through each time, and then the last step is going to be to wash that column with PE buffer that has ethanol in it. Okay, so get the PE bottle out of the kit that has the check on the top that says, yes, ethanol was added. All right? So you're going to get that out, and then you're going to add 0.75 milliliters of that PE buffer, and you're going to spin that down. Again, you're going to discard the flow through, but you're going to put that column back into the little tube and spin it down one more time. Right? It needs to be done twice in order to get all of that ethanol out of there. You want to get all the ethanol out of your little column. Okay? All right, and after you've done that, the 2 spins, all right, spin down, discard flow through, and spin down again. Okay, so you spin it twice with the PE buffer. All right, only add PE buffer once, spin it twice. Okay, then replace the clear tube with a clean Eppendorf tube. This is very important, because that tube that you spun down that ethanol buffer into has ethanol in it. You don't want ethanol. You want a nice, piece of DNA in EB buffer. Because after that, you want to be able to use enzymes on it. If you've got ethanol in there, it will denature the enzymes. Your enzymes won't cut anything. Okay? So we're going to put a clean Eppendorf tube under the little purple column, and then we're going to add, carefully, we're going to add 20 microliters. Don't follow how many the liquid it tells you to put in from the QIAGEN kit. It says add 50 microliters, or maybe 30 microliters. Either way, it's too much. Okay? So what we're going to do is we're going to only do 20 microliters, and the reason is so that we get a very concentrated PCR product out, and we're going to cut that whole thing with the restriction enzymes. Okay? So just add 20 microliters carefully. Drop it right down, bing, 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 into that little purple column with the white membrane. You want it right to land right on that membrane when you drop those 20 microliters of EB buffer. The EB buffer you find also in the QIAGEN kit. Okay, all these things come with the little bottles. They're all set up for you. Add it on there. Wait 4 to 5 minutes, because what's happening there is going to desorb, but if it soaks, it needs time to diffuse. And if it diffuses away into the buffer, okay, as it desorbs, then when you spin it down, boom, all that DNA comes out into the bottom, right? It works really well. Okay. So spin it down 1 minute when you've waited 4 to 5 minutes. 4 to 5, not 45. Okay. Spin down 1 minute and label your tube insert. All right. Next, we are going to do the restriction cut. All right. So the restriction cut, we need to use restriction enzymes. Restriction enzymes are one of the most expensive things that we use in the entire lab class. Okay. This teeny weeny little itty bitty tube here costs $70. Okay, and that has to be enough for all the students and then some. Okay, so this is what this little tube of stuff is for. So, and these enzymes are very sensitive. Okay, so I store them in the freezer in this blue box. You'll see it when you get there. Okay, a little handle on it. Carry the little handle with a little lid over. Put it on the counter, and when you're really ready to use those enzymes, that's when you take it out of the freezer. Then you open it, and you can see I leave the tube of enzyme in. There, it's got glycerol in it, so it's a liquid. You don't need to thaw it. Everything else you need to thaw, they're designed